All right, here's the thing about Rubisco. It kind of sucks. If you ask a botanist and especially an evolutionary biologist, somebody who knows plants, about Rubisco, they'll tell you it's a really, really inefficient enzyme. It's also just not great at doing its job correctly all the time because it can bind to CO2 or it can bind to oxygen gas. Like what? No, that's not what you're supposed to do, but that's what it does. If there's too much oxygen built up inside the cell, Rubisco's like, oh, cool, O2, it's like CO2. I can fix that. What? Why would you do that? Rubisco doesn't know, okay? It just binds to whatever fits. CO2 fits, oxygen fits, whatever. Okay, so plants in particular, we are now talking about plants and not photosynthetic algae, okay? So plants have pores in their leaves called stomata that open and close in response to heat and the amount of moisture in the air and things like that. When it's really hot, the plants close those pores in order to prevent water loss because they will lose water through dehydration. When that happens, O2 builds up inside the leaves, Rubisco binds to oxygen, fixes the oxygen, produces carbon dioxide as a waste product, and then just completely wastes a bunch of ATP that it's made. What? Why? What? Oh my gosh, Rubisco, what are you even doing? Now, evolutionarily, this makes sense if it evolved in a low oxygen environment, blah, blah, blah. Evolution does not come up with the perfect solution. It comes up with the solution that works and then keeps using it. This is one of the ones where it's not so perfect for the environment anymore, but it works. All right. So most plants don't deal with hot and dry temperatures and or conditions very often. They make enough sugars that it's not a big deal, and it's just they don't worry about it. This process, by the way, is called photorespiration when Rubisco binds to oxygen instead of CO2. So most plants, we call them C3 plants because they make a 3-carbon sugar at the end of or in the Calvin cycle. Everything just proceeds normally. That's like 85 to 90% of plants on Earth because 85 to 90% of plants on Earth didn't evolve in places like deserts because deserts are super harsh, dry environments. So plants that evolved in very hot and very dry conditions actually separate the light-dependent and the light-independent reactions, either in time or space, so that Rubisco can't bind to oxygen when oxygen builds up. I know. Okay, so there are two types that do things differently. There's C4 plants and CAM or CAM plants. The C4 plants actually separate the light-dependent reactions and the Calvin cycle in different cells in the leaf. So here's C3, carbon dioxide goes in, binds with Rubisco, Calvin cycle, blah, blah, blah. Water is lost when this pore is open on the cell. This is the stoma. Stoma is singular, stomata is plural. All right, C4 cells, carbon dioxide goes in, it diffuses into these mesophyll cells, which are the main ones that do the light reactions. And an enzyme makes that CO2 into a 4-carbon molecule, see now C4, 4 carbons, called oxaloacetate, which gets turned into malate, which gets broken up in different cells called the bundle sheath cells into CO2. These mesophyll cells are in most of the leaf of the plant. These bundle sheath cells are around the veins in the leaf of the plant. Okay, so these mesophyll cells don't have Rubisco. They literally can't do the Calvin cycle. So then the 4-carbon molecule needs to get moved, transported to the bundle sheath cells. 1 carbon split off, the other 3 go back as pyruvate into the mesophyll cells. La, 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 la. We keep dumping off carbon to bind with Rubisco to go through the Calvin cycle. Okay, so we literally separate light reactions and dark reactions in different cells in C4 plants. And the 4 is because they use a 4-carbon molecule rather than just the 3-carbon molecule going straight into the Calvin cycle. All right, the other set are the CAM plants, which means crassulacean acid metabolism. Crassulian refers to the group of plants where this was discovered, the Crassulidae, which I think includes like pineapples. And in these plants, the light-dependent reactions happen when the light is shining, and the light-independent reactions actually happen when the plant's stomata are open at night. So in order to separate those two, what it does is it makes a carbon molecule and stores it in a vacuole inside the cell. And then when the stomata open, and carbon dioxide is allowed to diffuse in, oxygen can freely diffuse out, then those vacuoles give up those carbon molecules so that the carbon can then bind in the Calvin cycle and do its thing. Okay, and then the Calvin cycle proceeds normally. So those are the two exceptions, just very few plants. Some cacti do this. I think a lot of like grasses, I think, that do C4, grasses in really dry places. So, but most plants, all your house plants, most crop plants are C3 plants, okay? So C4, 4-carbon molecule, separating light and dark reactions in space. CAM plants, separating light and dark reactions in time. And now you know everything you need to know about photosynthesis so you can be a photosynthesist.